The overall goal of this procedure is to introduce mRNA encoding a fluorescent protein into Branchiostoma lanceolatum oocytes to allow in vivo fluorescent imaging of developing embryos. This is accomplished by first inducing spawning of ripe Amphioxus males and females by a temperature shock of 24 hours. Following the temperature shock, females and males are placed into individual spawning cups, allowing the collection of, respectively, oocytes and sperm. Once the oocytes are lined up in a polylysine-coated dish, a specially prepared microinjection needle is used to inject solution inside the core of the oocyte. The final step is the fertilization of the oocyte with 1 to 5 drops of sperm. Ultimately, 2-photon laser scanning microscopy is used to show the ubiquitous expression of the fluorescent proteins produced from the microinjected mRNA.
Visual demonstration of this method is critical, as the microinjection steps are difficult to learn, because they require technical skills that are impossible to detail and convey in print.
To begin, prepare the polylysine-coated dishes used to immobilize the oocytes during the injection. For each 35 mm cell culture petri dish, cover the bottom with polylysine solution and incubate at room temperature for 5 minutes. Next, transfer the polylysine solution into another dish and again incubate at room temperature for 5 minutes. Discard the solution and let the petri dishes dry upside down at room temperature for 2 hours. When dry, store the coated dishes wrapped in a plastic wrap at 4 degrees Celsius.